Hi, welcome to this tutorial on probabilistic functional modes. My name is Rezvan Farahi-Bozorg, and this is part 3 of 3 lectures on this topic. In the previous lectures on probabilistic functional modes or PFMs, we learned about this framework and how it characterizes the brain networks in population and individuals from fMRI data. We also examined network matrix modeling in PFMs and compared the model to independent component analysis. In this third and last part of the lectures, we will focus on the latest version of PFMs, which is PFMs for big data, and we'll have a look at some of the key applications of the model to date. In recent years, there has been a massive growth in the availability of larger scale population neuroimaging studies. For example, UK Biobank is expected to include neuroimaging data from 100,000 participants. This type of large neuroimaging data are expected to provide rich resources that can enable us to map the brain function and disorders beyond what has been possible using conventional neuroimaging data. For example, by focusing on the fMRI data, we can aim to characterize the brain networks in population and individuals. These large population data sets typically examine different parts of subpopulation. For example, some of them might be focusing on aging groups or developmental babies or adolescents or even different patient groups. Therefore, a key characteristic of this type of data is that the population is not homogeneous. Therefore, we need models that can accommodate and can account for this type of heterogeneity in the data. For example, patients can be very different from each other, and they can also be very different from the group average. In the previous lectures on PFMs, we learned that these frameworks build a hierarchy between the population and individuals and simultaneously models the population and every individual subject inside the population. So it is specifically designed to accommodate large heterogeneity in the population, and therefore a key usefulness of a model like this would be for big data sets. So the latest version of the PFM framework, which is called Stochastic Probabilistic Functional Modes, was recently introduced to enable modeling big data populations as well as every individual inside these populations. Before we have a look at the characteristics of the stochastic PFMs, let's see why we need this new version of the model. In the previous lecture parts, we learned that the classic PFM framework, or classic PROFUMO, examines the population and all the subjects inside the population simultaneously, and it needs to have access to all the subjects in the data sets in order to reach a convergence and optimize the networks for the population and individuals simultaneously. However, because of this, as the number of subjects grows, for example, as we go over 2,000 or 3,000 subjects, the model starts becoming computationally very expensive and quickly becomes intractable. Therefore, the new version, the stochastic PROFUMO or stochastic PFMs, were proposed so that we can analyze large data sets, for example, 100,000 subjects in the UK Biobank, easily using the available or even the modest computing clusters. Now, let's see how the classic and stochastic PFMs are similar and how they are different. First, focusing on the similarities. Similar to the classic PFMs, we build a hierarchy between the population and individuals with two levels, group and individuals, where group level is used for top-down regularization of individual-specific networks, and evidence across individuals is accumulated to update the group. We iterate between these two levels of inference until convergence. Additionally, similarly, we build this hierarchy using Bayesian techniques. So, through Bayesian priors, we link the population and individuals together. And we built this hierarchy on two key elements of the model. First, the spatial topographies of the modes, the way that the functional modes are laid out across the brain voxels, and second, the temporal connectivity between the modes, so the functional and temporal interactions between the modes. The two versions of the model have also some key differences. The main difference is that in the stochastic PFMs, we cannot have access to the entire population. Instead, we want to build a model that can visit a small fraction of population in each of the model iterations and still yield accurate results both for the group and for every individual inside the group. In order to do this, the core idea is very simple. We divide subjects into small batches. For example, if we have 5,000 subjects, we can, in each of these batches, we can visit 50 of them, which is a small and very manageable number. And so in each of the model iterations, only this batch will receive input from the group and send the evidence back to the group. However, in order to make sure that this works, we need to take several things into account. Firstly, we want the group model to remain representative of the entire population and not just small batches that it visits at times. Therefore, we treat group level parameters as global parameters. These are continuously getting updated over time, while the subject batches of subjects are our local parameters that are visited in random intervals. Additionally, to keep the group model representative of the population, the group updates keep a memory of all the previous times that we have updated the model. The other thing that we need to take into account is that we want subjects to remain aligned with the group regardless of when they have been visited. For example, some of the subjects might have been visited during early batches where the group model is not yet a very accurate group model. We still want these subjects to be aligned with the latest and most updated version of the group, and therefore once the model reaches convergence, we revisit the entire population, each of the subjects, once or a few times to bring them up to speed with the latest version of the group. The third important thing is that we want the model to converge not only for the population but also for the individual subjects that we have visited in random intervals. And in order to enable that, in addition to the group model keeping a memory of its past, each subject also keeps a memory of its past. So every time we revisit a subject, instead of building its model from scratch, we will build it on the memory of the previous time that the subject was visited and was being updated. Another key difference between the stochastic and classic PFMs is that in lecture part 1, we described that classic PFMs use a technique called variational Bayesian inference to do the optimization of the entire framework. Here, we replace this with a technique called a stochastic variational Bayesian inference, which can enable us to visit a small fraction of the population in each of these model iterations and still reach a convergence. By taking these into account and by making these changes, the key aim of the model, as I said, is to reduce the computational costs, but also the model can be expected to accommodate perhaps slightly more population heterogeneity, because instead of visiting subjects in all model iterations, we only visit each subject in random intervals. So there's naturally slightly less push towards the group as we update the subjects over time. As an output from the model, similar to before, we will get spatial topographies and functional connectivities of the modes for the group and also for every individual. Okay, so far in all the parts of PFM lectures, we learned about the characteristics of the modeling framework itself and how to interpret different types of the results that we get from the model. In this last part, I will focus on some of the applications of the framework to date. The first application was that by applying the PFMs to a big data set, we were able to map the brain function or map the brain networks with more detail than has been possible using smaller populations. Here are the results of reconstructing 150 PFMs based on 5,000 subjects in UK Biobank. This can provide us with a high-dimensional soft parcellation of the brain. It is high-dimensional because the number is high, and it is a soft. It is called a soft parcellation because, similar to ICA, for example, the boundaries between the networks, which are the parcels in this framework, the boundaries are soft boundaries. They're not hard boundaries. So it is the existence of the modes is not binary in different brain voxels, but we can have soft boundaries between the modes in this type of parcellation. By extracting these high-dimensional PFMs, the PFMs show a really interesting behavior. So at lower dimensions, in the previous lecture parts, we saw that at lower dimensions, for example 25 or 50, we typically expect to get distributed larger scale functional modes that are spread around multiple brain regions that can be distant from each other. For example, the modes shown in the top row. So the ones that are shown in yellow boxes, these are the modes that we expect to get at lower dimensions. And the interesting behavior that PFM shows is that as we go to higher dimension, it keeps these distributed modes more or less intact, but it keeps adding more and more fine-grained modes in the brain. These include both fine-grained distributed modes, which are shown in blue boxes, and also parse-like modes that are contiguous and constrained to one brain region. One of the key differences between the modes that we found at lower dimensions and the ones that are added at higher dimensions is that the ones at lower dimensions typically have a higher signal-to-noise ratio, both spatially and temporally. If you remember from the previous ICA lectures and also from part 2 of the PFM lectures, this behavior is very different from what we expect and what we see in ICA, for example, spatial ICA. As we go to higher dimensions, the modes, the larger scale modes, start getting split up. But here with PFMs, there is no splitting happening because we can allow the modes to be spatially overlapping with each other. We maintain what we already discovered at lower dimensions, and newer modes keep getting added to the mix. Now let's examine the spatial and temporal correlations between these high-dimensional PFMs, so the NetMats. Here on the left, I'm showing the spatial correlation or spatial NetMat. That is a characterization of how much the modes overlap spatially with each other. As we can see, we discover 6 distinct clusters of modes based on their spatial overlap with each other. Now, if we look at the partial temporal correlation matrix or temporal NetMat, the architecture is less structured. However, the modes that are spatially more correlated with each other tend to be also more temporally correlated with each other. If you remember from lecture part 2, a key characteristic of the PFMs was that there were no restriction on spatial versus temporal independence between the modes. So we expect the model to end up somewhere between a spatial and temporal ICA. One interesting observation here is that at lower dimensions, we typically have a temporal correlation matrix that is largely unstructured, so the behavior of the model is slightly closer to the temporal ICA. But as we go to higher dimensions, we see that more temporal structure is also added to the temporal NetMats. So we move in the kind of more in the middle of the spatial and temporal ICA. Right, now let's move on to another application of the PFMs, another application that the PFM framework can have, particularly on big data. So a key goal of the neuroimaging is that by, particularly non-invasive neuroimaging, is that to enable us to examine images of the brain function or images of the brain and make predictions about individualistic traits, for example, age, cognitive scores, different disease scores, and so on and so forth. The individualistic modeling of functional networks in PFM frameworks it can be especially useful for us to try and make predictions about these individualistic traits based on how the brain networks are different across individuals. For this purpose, we can focus on the different elements of the model, for example, the spatial topographies of the modes, the spatial overlaps between the modes or spatial NetMats, and also temporal NetMats. We can also combine all of these elements, so that would be our predictors. Our target then in these predictions are the individualistic traits. For example, the examples that I will show in the next slide, we have used the cognitive scores obtained from UK Biobank as targets. And these can be different types of scores, like, for example, how well a person performed a memory function test, how well they did a reaction time test, executive function test, and so on and so forth. So we have now a prediction pipeline where PFM characteristics are extracted from PFMs are our predictors, and the cognitive tests are the targets that we want to make predictions about. We can use different approaches to do these predictions. The results that I will show have used elastic net regression with cross-validations to make these predictions. And finally, we can compare the PFM framework and the performance of the PFM framework to the more established ICA-Dual Regression paradigm. Here are kind of the raw results. So we categorize the cognitive tests into three categories: sensory and motor function, memory and executive function, and a range of fluid intelligence tests. And the PFMs were best when trying to predict memory and executive function tests, the outcome of memory and executive function tests in UK Biobank. Now let's see which PFMs did the best job at doing these predictions. If you remember the high dimensional postulation that I showed to you earlier, most of these modes are among the ones in the yellow boxes, so the distributed larger scale modes. However, some of them are also the ones that we start seeing, the more fine-grained modes that we start seeing at higher dimensions. So even though the larger scale modes are generally a better, the variation of the larger scale modes across individuals is better predictor of the cognitive outcome in individuals, some of the fine-grained modes might also play a very important role. So some fine-grained modes in the brain and how they differ between one person to the other can have a crucial role in prediction of the cognitive outcome. Right, now let's see how the model's performance compares to ICA and dual regression paradigm. Here first, let's look at the output from the full network. So both spatial and temporal characteristics all combined together, and we try to make prediction of the cognitive scores. With PFM results shown on the y-axis and ICA-Dual Regression results shown on the x-axis. The red line here shows equal performance, so as we go higher, it means that the PFMs are doing better, and as we go lower, it means that the ICA-Dual Regression is doing better. And as we can see, the overall network is doing a slightly better job, or a significantly better job for the PFMs compared to the ICA-Dual Regression. Now let's focus on the spatial characteristics and temporal characteristics of the modes separately. First, focusing on the spatial characteristics, so we can look at how well the variations of the spatial topographies of the modes can make predictions about the cognitive outcome in individual subjects. And also we can have a look at how variations of the spatial overlaps between the modes across individuals do when we try to make predictions about the cognitive outcome. For both of these scenarios, these spatial variations in PFMs are doing a significantly better job at making predictions about the cognitive outcome compared to ICA-Dual Regression. However, now if we look at the temporal correlations or the functional connectivity, the pattern reverses. So ICA-Dual Regression does a better job compared to the PFM. This is a very interesting result, so that we see a double dissociation. The spatial characteristics are better for the PFMs, the temporal characteristics are better for the ICA. This can be linked to what we already discussed extensively in PFM lecture part 2, where we were discussing that if the spatial variability across individuals and the spatial overlap between the modes are not accurately accounted for, this can get entangled and result into a biased estimation of temporal correlations, which is also the behavior that we see for the ICA-Dual Regression here. So in terms of interpretability, it is likely that what PFMs are measuring using spatial variations might be a better reflection of true sources of subject variability. Okay, now let's move on to the last part of this talk. So far, we were looking at predicting non-imaging variables using the functional networks. In this last part, we want to do something slightly different and make predictions about each person's brain response to different cognitive tasks based on the resting state PFMs. So each person's brain responds uniquely to cognitive tasks. For example, if we give them tasks related to emotion processing, working memory, or have them listen to stories, different people's brains respond differently to this task. A few years ago, a very exciting study was published that showed that the individualistic modeling of resting state networks, where people are doing no tasks, can predict how each person's brain responds uniquely to the tasks. Recently, Jung and colleagues showed that if we use PFMs to make predictions about these unique task responses based on resting state data, it improves our ability to make these predictions compared to the standard techniques. Okay, let's summarize what we learned in this lecture. Firstly, we learned about the latest version of PFMs, which is called Stochastic Probabilistic Punctional Modes, that enables us to obtain individualistic brain function mapping for larger scale population datasets and every individual inside those populations. Based on the application of PFMs to big data, we were able to obtain high-dimensional PFMs, which provide a fine-grained parcellation of the brain and show unique characteristics. And finally, we showed that the individualistic modeling in the PFM framework can improve our ability to make prediction about the behavioral outcome in people, and also to make predictions about how each person's brain responds uniquely when they are doing cognitive tasks. This is it for lecture part 3 and all 3 lectures of the PFM framework, and thank you very much for listening.